So answering the question, when we sort, are the chromosomes going to sort together or are they going to sort independently? In other words, when we take a heterozygous adult for two traits, are the capital letters going to always separate together and the lowercase together, or can we take those two combinations and divide them up any of the four ways? And so basically to figure that out, we just need to act it out and see what our gametes are going to look like. In other words, we're going to see which system explains things. Which of the actual results that Mendel got, which one does it support? And so, if we say, okay, well, let's take that first scenario. See if this is assuming that they travel together, right? That they are traveling together. We're going to get a capital R and a capital Y from one parent, capital R and capital Y from the other parent. Remember, this is the F1 generation that's being crossed to produce the F2 generation. Well, if that was the case, we would get big R, big Y, big Y, big R, big Y, little R, just like the parent. Another copy of that, big Y, little, big R, little, just like the parent. And then we would get all little letters, little y, little R, little R. And if this was the case, we would end up with a round yellow seed, a round yellow seed, a round yellow seed, and a wrinkled green seed. Again, just like the parents, right? Remember, or the grandparents, I should say. Remember the parent generation, one was round and yellow and one was wrinkled and green. So the question is that what's going to happen? Well, if we look at Mendel's results, he got yellow round, and he did get an occasional wrinkled green, but he also got a green round and a yellow wrinkled. So, based on those results and the prediction of our Punnett square, that does not seem like it can happen. And so if we look at the other situation, say, okay, well, what if they travel independently? Well, then we have to come up with our parent gametes. Now, can someone remind us how, we just kind of jumped ahead in this picture, how do we go from a parent of being a big Y, big R, little r, to those four different gamete combinations up there? Okay, but how do we do it?
Oh, yeah, like FOIL.
FOIL. Okay, so what does the F stand for in FOIL? First. All right, so we're doing FOIL. So we have first, which means the first of each letter, right? So the first Y with the first R, so we get capital Y, capital R. That's our first combination for a gamete. So we wrote it on the top. What does O stand for? Outside. So kind of like the bookends, if you think about these. You got the outside Y, and you got the outside R, and so that gives us big Y, little r. Big Y, little r. Then you got I standing for inside, so these two are trapped between the other two, and so we get little y and big R, so we wrote that on our third box, little y, big R. And then finally L stands for the last, which is the last Y with the last R. So the second y is lowercase, the second r is lowercase, so little y, little r. So all you got to do is FOIL the parent's genotype. Well, that gives us our answers across the top for the dad. The mom has the exact same genotype, so rather than having to FOIL again, we know it's just going to be the same combination. And so we write her along the left side. So now that we have the gametes, and again, you can think of it from a non-FOIL aspect, you should really kind of understand what's happening, but remember that as these parents are making their sex cell, it's basically a 50-50 chance. Is this Y going to go, this Y is going to go to half the sex cell, and this Y is going to go to half the sex cells, right? And this R is going to go to half the sex cells, and this R is going to go to half the sex cells. And that's basically what we were doing as we were FOILing. And so you can see half the sex cells of dad have a capital Y and half have a lowercase y, and half have a capital R and half have a lowercase r, right? And that's what gives us those four combinations. So now it's just a matter of filling things in. So we get all capitals, we get capital Y's and heterozygous R's, heterozygous Y's, capital R's, heterozygous for both. There's a couple repeating. There's a new one, 2 capitals and 2 lowercases. And so now we're just dropping the number or the letters down, dragging the letters over. Right? Hopefully you remember doing this over and over and over again sophomore year. A couple general rules as we do this. Remember the capital letter of a pair always comes first, followed by the lowercase letter, because if you sometimes write it one way and sometimes write it the other, you'll get very, very confused as it'll be hard. If one time you wrote this as little y, big R, big R, and over here you wrote it as big Y, big R, it's going to be hard to see that those two are the same thing, right? So always the capital letter first, always the lowercase. Whatever the parents were, if the parents were listed the Y's first and then the R's, you're going to list the Y's first and then the R's for the offspring. So pretty straightforward. At the very end here, we get all of our little letters. And so now it's time to actually have to analyze it, right? And so we have capital Y and capital R, so that's going to be both dominant traits, so that's going to be yellow and round. That one's 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 going to be yellow and wrinkled. Yellow and round. Yellow and wrinkled. Yellow and round. Yellow and round. Green and round.
Green and round.
Yellow and round. Yellow and wrinkled.
Wrinkled and round.
Green and round. And green and wrinkled. Right. So obviously a little bit of tedious and a little bit time consuming, but now we have these, and so we start to add them up, and we say, okay, so I got 1, 2, 3, 4, 5, 6, 7, 8, 9 that are yellow and round. I have 1, 2, 3 that are yellow and wrinkled, 1, 2, 3 that are round and green, and 1 out of 16 that is wrinkled and green, heterozygous for both traits. And compared to Mendel's results, remember he's doing this not just 16 times, but thousands of times like we saw in that earlier data table, and over again he was getting this 9/16, 3/16, 3/16, 1/16 ratio, right? Obviously not exactly that, but really close to that, right? Despite the different crosses that he did. And so this idea of the genes being independent, right, being able to make all these different combinations was supported, and this mechanism works compared to the results that Mendel was getting. Yes?
When you see the results like that, how do you write the ratio? Like, you know how when we were doing the volume where it was like 9 to 1, how do you like do the ratio for those? Is it 9 to 3 to 3 to 1? And
So you would go 9, 3, 3, 1.
Okay, so you just
Probably
don't
right, too.
take out you just don't use the 16, like you don't have to divide them out or anything.
Right. You don't necessarily have to write it ratios, but this would be 9, 3, 3 to 1 would be the technical ratio. You could say 9/16, 3/16, 3/16, and 1/16. You could say 56%, 18%, 18%, 6%.
Okay.
And again, these would be the phenotype ratios, right? It gets a little bit more complex. We only have 4 phenotypes, but we have a lot more genotypes. I think if we looked at that, we'd have 9 genotypes, 9 different genotypes that lead to 4 different phenotypes. And so remember, you got to think about what you have to report as well when you read a question. Is it asking me for phenotypes? Is it asking me for genotypes? Is it asking me for both? And kind of starting tomorrow in early hour, we'll take this idea of kind of taking the predicted from a Punnett square and actually taking real data. And then the question is, okay, does my real data agree with these numbers? Does my real data agree with those monohybrid numbers of 3 to 1, 75%, 25%? And so we'll do a statistical test tomorrow to figure out, okay, so how do I know if my results are significantly different or if they're significantly similar? So Mendel's second law, remember his first law was that segregation, or that separation, right? So his second law deals with his independent assortment, that different genes, all right, different loci, different place on the genes, on the chromosomes, separate into gametes independently. So different genes will separate into gametes independently. And so if we follow this through, we've got a yellow chromosome and a green, we've got round, and we have wrinkled. So there's our 2 different traits, right? Just 2 options for each trait, so it's still Mendelian genetics. You have the dominant and the recessive. And so here we can see the parent, based on our little key over there. This parent has the yellow gene, and the green gene, and the round gene, and the wrinkled gene, right? So they got the nice sampler platter of genes in there. And so, as they undergo meiosis, those genes are going to double, right? What's my diploid number in this cell, by the way? 4, right? 1, 2, 3, 4. So now we're forming 4 X's. We're forming 2 tetrads, it looks like there. Remember, you always form the number of tetrads as a haploid cell of that. And so we've got our homologous pairs of yellow and green hooking up. We've got our homologous pair of round versus wrinkled hooking up. Some crossing over might occur. And so when we work this all out and actually go through all of meiosis, we end up with some different combinations here. Here we get a big R, little r, and it'd probably be better if we had more room to actually just go through this whole thing. But remember, the homologous pairs are going to get separated, right? The tetrads are going to split into one, and these tetrads are going to split. So one cell is going to end up with a yellow X and the wavy guy. The other cell is going to end up with a green X and the non-wavy guy. And then those will separate to form two identical cells. And so that's why we get, because there was no crossing over going on that we track, that's why these two cells look the same and these two cells look the same. But we can clearly see when we do this that each cell is going to get one copy of the chromosome that had the color, and each cell is going to get one of the bigger chromosomes that had the round versus wrinkle. And so we end up with those four combinations, just like we were doing before. And so here is our other parent. Come on. There we go. And so our parents, we basically just FOIL them. And ultimately what that is showing you is, again, the independent assortment. Can someone remind me, where does that independent assortment happen? Metaphase, good. A little bit more specific though. Metaphase 1 of meiosis. Good. Remember when you were doing those poppet beads, we said, okay, so they're kind of, these guys are just lining up across the middle, right? And it's just a matter of chance when these lined up, there was a matter of chance is, does this go this way, or could these flip the other way and have the yellow on the right? And likewise, these could go this way, or those could flip. And so I have two possibilities, two things with two possibilities, and so that's why there are four different ways that that could act itself out. So the non-homologous chromosomes, and that's an important key that we'll kind of come back to in 8.2. It's the non-homologous chromosomes aligned independently. Classes of gametes produced in equal amounts. And so again, let's see here. Okay, so in the equal amounts is the idea, right? When we did this, we got the equal amounts of the gametes, right? 1 to 1 to 1 to 1 ratio. And it's only true for genes on separate chromosomes. And here's where we're going to have to clarify things a little bit tomorrow probably. Only true for genes on separate chromosomes or on the same chromosome, but allowed crossing over to happen. Because when you think about it, if this gene for yellow and if the gene for round were on the same chromosome, that chromosome is going to travel together, right, as one thing. And so this, and what Mendel's plants were doing, is he was looking at traits that were on different chromosomes. And those different chromosomes did separate independently. What we'll find out as we kind of continue on this chapter is sometimes we have genes on we obviously have genes on the same chromosome, right? Each chromosome has about 1,000 genes. And so if that gene is on chromosome 1 and the other gene is a little bit further down on chromosome 1, odds are they're going to travel together, and then you would not see this independent assortment. Unless by chance crossing over happened on that part to separate those two genes. And we'll clarify that a little bit more in 8.2, and that's the focus of it. So which stage of the law creates this independent assortment? It's going to be in metaphase 1, when it's a random chance on how those chromosomes line up across the middle. In other words, it's a random chance of which side of the tetrad is going to face which direction. So in this case, both the red ones were facing the left and both blue ones were facing the right. In a different situation, both blue ones could be facing the left and red could be facing the right. Or this red one could stay this way and this one could have been switched. Or this one could have stayed this way and this one could have been switched. Right? And again, it's almost harder to see that with just, 4 chromosomes there. But when you start to figure out 23 tetrads lining up across the middle, and the fact that each tetrad could face one way or the other, right? And I could flip this one and keep the other 22 the same, or I could flip this one, or et cetera, et cetera. It gets kind of crazy in terms of those numbers, and that's where that number of about 8.4 million came in, right? In the previous chapter. And then 8.4 million from the sperm and 8.4 million of the egg gave us that 70 trillion different combinations of the zygote, right? And again, that's even ignoring, you can see that a little crossing over is actually happening there. So again, the exception, that again we'll clarify later on as well, is if the genes are on the same chromosome and close together. So if these are my two genes, if this was color and if this was texture, wrinkled versus round, odds are those are not going to travel separately, right? If I had a dominant trait for color, yellow, and a dominant trait for being round, well, if this had been Mendel's genetics, the round and the yellow would almost always travel together, right? Because they're right next to each other on the chromosome. And so sometimes we study traits and we're like, oh, that's independent assortment doesn't seem to be happening, right? These two traits always travel together, and the reason is because they're on the same chromosome. They're close enough that crossing over doesn't happen. The closer two genes are, the less times crossing over happens. Because think about it, the odds of crossing over happening right between them, the closer that distance is, the less the chance that they're going to split off between the middle of them, right? The farther they are apart on a chromosome, the better the chance that a crossing over event can happen between them, right? And so when they are on the same genes like that, we call them being linked, which is something that we'll talk about a little bit later on in this chapter. And so they usually be inherited together. Rarely will crossing over happen between those two genes that are too close to do that. So they are linked. So, once again, kind of a reminder that although we clearly see the chromosomes doing all this, and we can take that process of meiosis that we studied and tie it in with kind of the results that Mendel saw in these pea plants, you got to remember that Mendel wasn't familiar with DNA, right? That wasn't known to be the genetic material until 1950s. Didn't even use the word genes. He called them that these factors are passing on, right? Had no idea of the structural idea behind any of this stuff. And so to kind of take in Mendels laws and the idea of meiosis here, we've got our parents, so we can see their chromosomes. What's our diploid number of these cells? 4, and our haploid number? 2. And so we have our parent generation kind of skipping through meiosis and not really showing any of the steps, just saying, okay, so here's my parent, so here's my gamete, here's another parent, here are the two gametes. You can see diploid and haploid, diploid and haploid. Two of the haploid cells come together to form another diploid. Remember, this is just what he was doing. You had the true breeding yellow and the true breeding round with a true breeding wrinkled and a true breeding green. So all the offspring were heterozygous, big R, little y, little y. So now in metaphase, you can see our tetrads pairing up. 2 tetrads because we have a haploid number of 2. Again, independent assortment going on there. There's different arrangements, 4 different arrangements for each of these cells and how they could have done that. So now we separate the homologous pairs. We rip the tetrads apart, happening with both of our ideas here. So we end up with metaphase 2. Now we have to separate our sister chromatids, and we end up with all these different combinations. Well, we end up with our gametes. The biggest thing to see is that we have one of each chromosome, right? So they're haploid. We have one bigger one and one smaller one. And if we add up kind of our whole data here, we get a quarter of them being big R, big Y, a quarter of them being little r, little y, a quarter of them being little r, big Y, and a quarter of them being big R, little y, right? Just like what our foiling is going to give us, right? A 1 to 1 to 1 to 1 ratio, or a 1/4 or 25% each. And so now it's a matter of kind of chance, and that's what the Punnett square is going to do for us, right? It's a matter of chance of one of these meeting another one, right? 1 to the 4, 1/4 times 1/4 gives you 1/16. And so as we filled in that Punnett square earlier, we saw that we get this 9 to 3 to 3 to 1 ratio, having the most that are dominant and for both traits, the least being recessive for both traits, and then 6 of the combinations are heterozygous for, I should say, show 1 dominant trait and 1 of the recessive traits. So to review Mendel's laws, both very important. The law of segregation can be shown in a monohybrid cross, merely stating that a single trait, when that trait is separated, or when those gametes are formed, one allele has to go to each gamete cell being produced. So each allele segregates into separate gametes. Parent is big P, that means one P has to go to each gamete. If he's big P, little p, that means the big P is going to go to half the gametes, the little p is going to get separated and go into half the gametes. And this is where a lot of your meiosis failed on that 3rd page, right? When some of you had 2 big A's, or big A, little a, and 2 B's and 2 D's, right? You failed to segregate your chromosomes, right? They've got to separate, so you only have 1 of each copy. And that would be occurring in metaphase 1. The law of independent assortment deals with dihybrid or more, and there are trihybrids and beyond. So dihybrids and more. So this is the idea that as those tetrads are lining up during also metaphase 1, that genes on separate chromosomes are going to independently separate or segregate or sort into their own gametes. So just since the parent cell had 2 dominant alleles doesn't mean that they have to travel together. You can mix them up, and again, we get those by foiling. And again, the exception is when you have linked genes, which will kind of come up throughout the rest of this chapter. So finally, to kind of conclude, Mendel ended up, he chose pretty wisely as he went through, as he chose these pea plants. Now, there's several reasons why he ended up with pea plants. One was that they were already growing them at the monastery from the things I read. But pea plants, it turns out that if he had chosen some other organisms to study these things, he would not have discovered the trait, he would not have discovered the inheritance traits that he did. But some of the reasons why these pea plants worked out well is, first of all, they're available in many varieties, and they had some very clear characteristics, such as flower color, seed shape, and again, Mendel studied 7 different characteristics. We didn't talk about it much, but tall versus short was another very clear one. Also, he had strict control over which plants mated, right? You can just kind of snip on the anthers and take away the male's pollen, leaving only the stigma to receive pollen, so you can cross-pollinate. Similar to kind of what we forced our fast plants to do earlier, right? We prevented them from self-pollinating, and they're kind of genetically prevented anyways, but we kind of made sure that any crosses that were happening that we caused to happen. So each plant has a male and female structure. They can self-fertilize, but you can also prevent them from self-fertilizing, and he can cross-pollinate them. Also, they're relatively simple genetically. And so when you start to look at the genetics of them, most characters in a pea plant, this is not true of you guys, most characters in a pea plant are controlled by a single gene, with each gene having only 2 alleles. In other words, if you wonder why did that flower of that pea plant have a purple color, well, first of all, it's only due to 1 gene. And that 1 gene had 2 different alleles, 2 different possibilities, either the purple, right, making the purple protein, or not making the purple protein. And if you start to think about how your genetics go, and you start to say, well, okay, so let's study my height, and my height, you'd find, is not controlled by 1 gene. There's a lot of factors at play that control your height. And some of them are genetic, and some of them are environmental, and you're not going to get this simple dominant of recessive, right? And so it would be very hard. You won't be able to see this idea of these characteristics. And so, again, a very, very good choice that allowed him to kind of get the traits that he was able to get.